A very good day to all the honorable attendees. My name is Dayang Sharyati, Datu Abdul Salam, and today I'll be presenting my review paper on Nuclear-Factor-Kappa-B or NF-Kappa-B in Silico studies. I am a PhD candidate at Swinburne University of Technology. I am doing research on in silico nasopharyngeal carcinoma therapeutics via targeting the NF-Kappa-B signaling pathway. The agenda of my presentation today is as follows. I will start with the background of my study, its significance, NF-kappa-B protein, inflammatory and immune response, cancer, neurodegenerative, other diseases related to NF-kappa-B, future perspectives, the discussion, and the conclusion and takeaways from my presentation. We start with the background. NF-kappa-B is found in almost all animal cell types and plays a role in cellular responses to cytokines, stress, free radicals, heavy metals, ultraviolet irradiation, oxidized low-density lipoprotein, and bacteria or viral antigens. The transcription factor NF-kappa-B is necessary to control the immune response to infection. The NF-kappa-B malfunction has been linked to cancer, inflammatory and autoimmune diseases, septic shock, viral infection, and aberrant immunological development. Computer-aided or in silico studies on the NF-kappa-B complex give hope for developing potential therapeutic targets for various diseases involving the NF-kappa-B protein. In silico investigations, as opposed to in vivo and in vitro research, have the advantages of being more exact and time efficient and eliminating the utilization of humans and animals. It provides a framework for evaluating the activity of prospective therapies against molecular targets, predicting the activity based on a compound structure even before it has been synthesized for additional in vitro and in vivo research. Targeting NF-kappa-B protein is crucial as it can implicate many diseases such as immune and inflammatory responses involved in stress response, cellular growth, and apoptosis. These findings are critical for directing future research into the NF-kappa-B protein, its function in human disorders, and prospective therapeutic targets. The NF-kappa-B protein complex is typically found in the cytoplasm of cells and comprises of 5 proteins: P65 or REL-A, REL-B, CREL, P105, and P50, which is the NF-kappa-B1, and P100 and P52, which is the NF-kappa-B2. NF-kappa-B proteins can form up to 15 different dimers through combinatorial associations. The P50 and P65 heterodimer is the most abundant and found in almost all cell types. IKB proteins such as IKB-A bind to NF-kappa-B protein and inhibit the activity by masking the nuclear localization sequences. IKB-A is the best studied member of the IKB family and is critical for the regulation of the NF-kappa-B activity. Next, we shall look into the NF-kappa-B signaling pathway, which is generally divided into canonical, non-canonical, or atypical pathway. NF-kappa-B protein complex is released from the cytoplasm and translocated to the nucleus when cells are activated. In immunological response, inflammation, cell proliferation, and development, the NF-kappa-B complex binds to DNA and promotes gene transcription. The end product of the canonical and atypical NF-kappa-B pathway is the P50 and P65 heterodimer. Meanwhile, the P52 and REL-B is the complex generated through non-canonical pathway. As mentioned earlier, NF-kappa-B pathway dysregulation can cause inflammatory and autoimmune illnesses. When the NF-kappa-B canonical pathway is activated, pro-inflammatory cytokines such as ILK1, ILK6, and TNF are produced. The NF-kappa-B protein complex also modulates inflammatory T cell activation. Autoimmunity causes cell destruction and chronic inflammation due to a prolonged immunological response against self-antigens. Rheumatoid arthritis, inflammatory bowel disease, asthma, chronic obstructive lung disease, and diabetes are among the autoimmune and inflammatory disorders implicated by the NF-kappa-B signaling pathways. Cannon et al. Employed the quantitative structure-activity relationship models to test for anti-inflammation medicines that target the NF-kappa-B heterodimer complex as a potential COVID-19 therapeutic target. Apart from that, molecular docking experiments are used for comparative in vitro studies of NF-kappa-B complex as anti-type 2 diabetes mellitus compounds. For natural mechanical in walnuts, urolithin A and B, gallic acid, and ellagic acid show the ability to dynamically bind to the active site of NF-kappa-B with varied affinities. Meanwhile, Datu et al. Used a computer modeling technique to examine synovial tissue-based pharmaceutical targets and diagnostic biomarkers in rheumatoid arthritis. They discovered that inhibiting several prominent signaling pathways could potentially serve as drug targets by developing a mathematical model with 30 2-gene and 3-gene network. For many years, researchers have hypothesized a link between inflammation and cancer. The missing connection between these two processes might be NF-kappa-B. Inflammatory stimuli stimulate NF-kappa-B, and constitutive NF-kappa-B activity has been associated with cancer. NF-kappa-B activation can also contribute to developing leukemia and lymphoma. In myeloid and lymphoma cells, NF-kappa-B can be activated in response to growth factors and cytokines and the production of certain viral oncoproteins. Shankar et al. Present an intriguing computational analysis and in vitro investigation to evaluate the mechanical root components of the NF-kappa-B signaling pathways in prostate cancer. Their findings showed that an IKK complex component can coordinate the NF-kappa-B pathways. Apart from that, Murwanti et al. Performed molecular docking analysis to elucidate the binding poses of protein-ligand interactions between NF-kappa-B and curcumin in a study on triple-negative breast cancer. NF-kappa-B signaling pathway is also implicated in the neurological system, particularly in the cortex and hippocampus areas, which are essential in human learning and memory. Defects in NF-kappa-B signaling can produce pro-inflammatory mediators such as TNF-A, causing inflammation within the nervous system and leading to disorders such as Alzheimer's and Parkinson's. Hira et al. Research to weigh the efficacy of aldosterone antagonists, which is eplerenone, in a streptozotocin-induced Alzheimer's disease model. In silico modeling was used to examine the chemical behavior of substances that inhibit acetylcholinesterase using induced fit docking. Behavioral paradigms such as passive avoidance, race plasmase, Morris water maze, open field, and balancing beam were used to test Alzheimer's anti-impact. A lesser-known disease that could arise from the effect of pro-inflammatory cytokines generated due to dysregulation of the NF-kappa-B signaling pathway is depression. Depression is generally due to tissue damage resulting in an unpleasant sensory and emotional experience. Hence, seeking excellent and effective analgesics is vital for the treating this depression disorder. Stroke is another brain disease associated with the NF-kappa-B signaling pathway due to its irregularity inducing inflammation. The discovery of NF-kappa-B inhibitors is noteworthy because it provides a unique treatment method for various disorders, including inflammatory diseases and viral infections. Some of the tools included for experiment conducted by computer are data analysis, data mining, machine learning, and network analysis. In silico studies give hope for developing potential therapeutic targets for various diseases involving the NF-kappa-B signaling pathway. In summary, targeting the NF-kappa-B protein complex is crucial as it can implicate many diseases described previously. Before moving on to in vitro and in vivo investigation, in silico approaches were preferred for predicting NF-kappa-B targeted treatment. Apart from reducing the time and cost of research studies, the in silico experiment decreased the use of animal models and human cohorts, which is the highlight of the ethical research debate. Future NF-kappa-B protein complex studies should explore and adopt various ways, perhaps the in silico ways, to understand the complex nature of signaling pathways and their roles in numerous diseases. So before I end my presentation, here are some of the references which I used. And thank you so much for listening.